Hi, welcome to the Bio and AD Tutoring Center videos. The topic of today's video is cell cycle regulation. So by now you should have already learned about mitosis, or the process in which we divide a cell by copying its DNA and dividing that nucleus into two genetically identical daughter cells. But we don't want to be dividing cells all the time. We need to have some way to regulate that division, or else it can cause problems. And one of those primary problems is cancer. It's a result of cells dividing when they're not supposed to. And so every cell is different in the amount that it needs to divide. Some cells, such as skin cells, are divided constantly because we constantly need to replace them. Other cells, such as nerve cells, when they're mature and adults, will never divide, and that's why if you damage your nerves, it can be a problem. So the way that we control this is by the cell cycle control system. It involves three main checkpoints, or otherwise called restriction points. They need to be passed before they continue on the cell cycle. So there's one that's called the G1 checkpoint. It happens at the end of G1. There's one at G2, and there is the M checkpoint. So the G1 checkpoint checks to see if we want to divide. The cell says, hey, do we want to divide, or do we want to just stay being one single cell? And if it says yes, then it says, okay, we're going to replicate our DNA so we can go throughout this whole process. If it says, no, we don't want to divide, then it's going to say, okay, we're just going to go to G0 phase, which is a non-dividing phase, and stay there and not divide. And so that's what a lot of nerve cells are in, is G0. And actually a lot of cells in our body are in G0. They're not dividing anymore. The G2 checkpoint says, did we replicate the DNA correctly? So it's saying, before we actually divide this nucleus, let's make sure we got everything right so we don't have any mutations or errors. And so that allows the new daughter cell to be identically similar to the parent cell. And then the last checkpoint is the M checkpoint, and it occurs during metaphase, after all of the chromosomes are lined up along the metaphase plate. And it just asks, are all the chromosomes lined up properly? Is everything in its proper place? And are all the kinetochores attached to the chromosomes? Because if they're not, we're going to have some problems. And so if it passes that, then we'll go into anaphase, and we'll divide the nucleus, and then we'll divide the cytoskeleton. So those are the cell cycle control systems. So now we're going to get into a little bit more about which ones, ways that we can test and say yes or no to G1 and G2. So these first four all have to do with G1, and then this one right here has to do with G2. So a cell, when it's at the G1 checkpoint, it's deciding whether it's going to divide yet. And some of the things that determine that are if it's has enough nutrients. So it's nutrient dependent. That's not going to divide if it knows there's not enough nutrients to support the daughter cells. The second thing is a growth factor. So a lot of cells actually need a signal to be told to divide, and we talked a lot about cell signals in 3 separate videos, and so they need something to actually bind to a receptor molecule and link to some pathway to say, okay, you need to divide now. The next one is that they're density dependent. So cell is not going to divide if there is a lot of other cells around it and next to it, because that's going to cause some problems, and it doesn't like being squished. The last one is anchorage dependent, which just means that a cell needs to be connected to another cell or something else in order to divide. And then this is the mitosis promoting factor that we're going to talk about, and it's more of a specific chemical way in which the G2 checkpoint is overcome. So the mitosis promoting factor is made up of CDK and a cyclin. CDK stands for cyclin dependent kinase. It's basically a kinase that is dependent on a cyclin to work. Very simple, it matches its name. And a cyclin is just a molecule that is called a cyclin because it is circular in when it is around and when it's not. It goes through a cycle. So during G1, we have our CDKs. We have our CDKs throughout the entirety of this. We don't have a lot of cyclin. And as we go on, we start to slowly build up cyclin. We build up enough cyclin that it attaches to the CDK, and it will combine to form the MPF, mitosis promoting factor. And that mitosis promoting factor is the thing that needs to be present in order for the G2 checkpoint to be overcome. So the mitosis promoting factor will pass the G2 checkpoint, and then the cyclin will be degraded into these little pieces. and then it will continue on again. And so that's the mitosis promoting factor. Probably will have some questions on tests about it. Just know it's made up of cyclin and CDK, and it helps overcome the G2 checkpoint. That's all for today's video. In another video, I'm going to continue on with this and talk more about cell cycle regulation and how it can lead to cancer. Thank you for watching.